Good morning, dear students. Do you remember what we learned yesterday? We learned about the process of crystallization. And what was crystallization? It is a phenomena in which when a supersaturated solution is cooled down after further heating, it is left for cooling. Then what happens? Some crystals of that particular substance are formed. So yesterday we learned it theoretically. Today we are going to perform or learn with the help of an activity in which we are going to prepare the crystals of sugar by crystallization. This you can do at home also, but don't forget that it has to be done under the supervision of some adult or someone who is elder than you. Okay, like your mother, father, grandfather or grandmother. Okay, not like you are just a grown-up sibling of yours. Okay, you have to be under the strict supervision of some adult person in your house. So let us see what is it. We are going to prepare the crystals of sugar by technique of or by the method of crystallization. So what are the materials required for this? We require some, first of all, what do we require? Table sugar. Material required. Okay, this is an experiment in your lab manual also. So we are going to start from here first. What are the things that are required for this experiment? Table sugar or sugar that you use in your houses for that is an edible substance which gives sweet taste to your edible things that you make. So distilled water, you know what is distilled water? Distilled water is the purest form of water. Distilled water. Then what else? A beaker, a burner, beaker, burner. This we are talking when we are doing the work in lab. Okay. Then some stand, a cotton thread, cotton thread is required, a filter paper, okay. What else is required? Glass funnel and a glass rod. Glass funnel and a glass rod. Okay, these are the things that are required by us. Let us see again. Sugar, distilled water, beaker, burner, stand, cotton thread, filter paper, glass funnel and glass rod. Okay, now what is the procedure? How to perform this activity? What we will take in the initial step, first we are going to take about 100 ml of water in a beaker. Okay, in a beaker. See, this is the beaker, like this, a glass container which is used in the laboratory experiments. Okay, 100 ml of water is taken into the beaker. Now we are going to dissolve the sugar in the water till it makes a saturated solution. You know what is saturated solution? Saturated, there are two terms I am going to use. One is saturated and one is supersaturated. So what is saturated? Saturated solution is a type of solution in which no more, like here we are taking sugar. So I am going to talk about sugar, in which no more sugar can be added at a particular temperature. Say at room temperature. Room temperature means whatever the conditions are at this time of temperature, that. So what we will do? We will take beaker, 100 ml of water and put sugar in it, and with the help of glass rod we are going to dissolve it, stir it. So when this sugar is dissolved, then what we are going to do? We will put some more sugar in it. Okay, and we are not going to heat or doing anything. We are just adding sugar and stirring, adding sugar and stirring. A time will come when the water is not going to dissolve any more sugar in it. Okay, then this solution will be called a saturated solution. What do we call it? Saturated solution. Now what we will do, we are going to boil this saturated solution. Okay, we are going to keep it on a burner and boil the solution. Now when the solution is boiled, okay, we will add some more sugar in it in very small quantities, and we'll keep on stirring with the help of what? Glass rod. Okay, glass rod is meant for this stirring purpose only. Now what you are going to see, that sugar will get dissolved. When the heat is there, it means the sugar will be now more dissolved in it. Now when a time will come when no more sugar can be dissolved in that boiling solution also. So during that time, at that time when no more sugar is being added to boiled solution also, we will remove the beaker from the burner. Okay, we are going to remove the beaker from the burner and care with lots of care. Okay, we have to be very much careful. Why? Because we are dealing with the fire here, with the hot solution. It can fall on the body and can cause severe burns. So to avoid that, somebody should be there to help you. Okay, now when this has been removed, this beaker has been removed from the burner, what we will do, we are going to just filter it. Okay, we will filter it. How to filter it? Now this will be done like funnel will be taken, in that we are going to put a filter paper. See, filter paper is round in shape. So what we will do, we will fold it first half. Half means it will become like a semicircle. Okay, it will become like a semicircle this way. Now one more half, one more fold, means 2 times we are going to fold it, and it will become like a pocket like structure. So we are going to put in the funnel. Funnel is what? Funnel is a glass apparatus which is in this shape. Okay, so this unfolding, it will become like this, the filter paper. We are going to put it here, and in another beaker we are going to collect the filtrate. Okay, we will leave it for cooling. So when we are going to leave it for cooling, with the help of another glass rod or stand, a thread, cotton thread is tied on this string, is tied on this stand and left undisturbed in this solution. Okay, I am again repeating. When we have filtered that supersaturated solution with this method, like we are going to put a funnel, in that funnel we will put the folded tissue, this paper, okay, filter paper, and then through this we are going to pour that supersaturated solution. We will get the filtrate over here. Now this filtrate, which is collected in the beaker, is kept undisturbed. Okay, before that what we will do, we will take one stand and will tie a cotton thread on it and suspend it and let it hang like this in the solution. Okay, in this beaker. Now we will keep it undisturbed as shown in the picture. The string should dip into that solution. Okay, now leave this solution with the suspended cotton thread in it for 2 hours and allow it to cool. Let it cool. So after 2 hours, what you are going to observe? You are going to see that the thread has some deposition on it. Okay, the crystals are formed over the thread. Fine. So what does this experiment tell you? It says that, what is the conclusion that you are going to conclude from this experiment? When a supersaturated solution cools down, the sugar particles that are present in it starts separating out. Okay, when this supersaturated solution is allowed to cool, then what happens? The crystals of this supersaturated solution, they start separating out, and these sugar particles, they aggregate or they collect, they form a collection. They start collecting in definite or in some geometrical shapes that form the crystals. Yesterday I told you, in last class I told you that the crystals can be of any shape, just like they can be tetrahedral, they can be cubical, they can be cuboidal, so they can be of any shape. Okay, so this is how we are going to perform the sugar, to prepare the sugar crystals by the method of crystallization. So one quick recap, what we are going to do, we will take 100 ml of water and dissolve sugar in it and make a saturated solution. So this solution, saturated solution, will be boiled. In this boiling solution, we are going to put some more sugar and keep on stirring. A time will come when this boiling solution will also not add the sugar in it. Okay, the sugar will not be dissolved in it. So at that time we are going to remove it from the Bunsen burner or the flame, and we will filter this solution. Okay, so that the things which are not required, they do not go in the filtrate. So after that, what we will do, we will place a stand and tie a thread on it and suspend it in such a way that the end of the thread is hanging in the, or it's dipped into the solution. Okay, and leave it undisturbed for 2 hours. For how many hours? 2 hours, and let it cool. After 2 hours, what we are going to observe, that some crystals, they start depositing on the thread. Okay, so this is how the practical or the experiment, practically we can prove that crystallization takes place. And this we can do with the help of salt also. Okay, it's not that only sugar is required, we can do it with the salt also. So today's work is to write this activity in your notebook and draw the diagrams to support the activity in which you prepare sugar crystals by crystallization.